A very good morning to all of you, my dear children. In this video, I would like to discuss Kingdom Monera under the chapter Biological Classification. In my previous video, I have discussed history of biological classification where we have seen different biologists considered different types of criteria for classifying the organisms. The most accepted one is the Whittaker's Five Kingdom classification, right? In this video and in my next video, I will discuss Kingdom Monera, Archaebacteria, Eubacteria, Cyanobacteria, Mycoplasma, right? Let's get started. Kingdom Monera. This kingdom includes the most ancient, smallest, simplest organisms. It is believed that when the earth has evolved, that time the organisms which have originated, they are the monerans, Archaebacteria. That time the environment was in extreme condition, high temperature, saline condition, but these monerans could survive because of their spatial structure. We will study. They occur almost everywhere where the other organisms cannot survive. They may be present inside the plants and animals or on its surface. Thus, they may be present as ectoparasite or endoparasite inside the organism. For example, Salmonella typhi, Vibrio cholerae. They are responsible for developing diseases. They may be present in the soil, can act as decomposers. For example, Clostridium. They may be present in the saline water, extreme salty areas, in the high depths of the ocean. They may be present in the extreme cold region or even extreme hot region where the temperature is high as 100 degrees centigrade, or saline condition where the pH is as 2 or maybe less than that. They are present in the environment where the other organisms cannot survive. Monerans are unicellular, filamentous, or colonial. Important feature is they are prokaryotic in nature. Do you know what is the meaning of prokaryotic? Pro means primitive. That means primitive type of organisms. Unicellular, without any nuclear membrane. DNA is circular, double-stranded, and without any nuclear membrane. Okay, this type of genetic material is known as nucleoid. Membrane-bound cell organelles like mitochondria, lysosome, plastids, Golgi bodies, endoplasmic reticulum are absent. Ribosome is of 70S type. Respiratory enzymes are present in the plasma membrane.
Monerans lack a definite nucleus, but the nuclear material consists of a point-naked strand of DNA which is not enclosed in the nuclear membrane.
The cell wall is very unique. Cell wall is rigid, made up of peptidoglycan layer. Except Archaebacteria and the Mycoplasma. Mycoplasma do not possess cell wall. Bacteria surrounded by two protective coverings, inner cell membrane and then outer cell wall. Outside the cell wall, a covering is present that is called the capsule. Short whip-like extensions, pili, surround the surface of the bacteria, which helps in conjugation. And the locomotory organ is the flagella, which may be one in number or maybe more in number, may be present at one end or maybe throughout its surface or maybe both sides. It is the locomotory organ, long whip-like extension. Now the next one is the mode of nutrition. How do they get energy? That is the most important thing. They are autotrophic or heterotropic. Autotrophic means they can make their own food, but the autotrophic monerans once again are of two types, that is photosynthetic autotrophs or maybe chemosynthetic autotrophs. Photosynthetic autotrophs means solar energy is utilized for fixing the carbon dioxide to form carbohydrate. Photosynthetic autotrophs once again are of two types, oxygenic and anoxygenic. Oxygenic photosynthetic autotrophs, what does it mean? That means during the process, oxygen is evolved. What happens, do you know? Here, the reducing power obtained from the hydrogen, and this hydrogen comes from the splitting of the water. Thus, in oxygenic photosynthesis, definitely the food is produced, carbohydrate is produced, but along with the carbohydrate, oxygen is also evolved. That is the oxygenic photosynthesis. Certain bacteria contains a special type of pigment, bacteriochlorophyll and bacterioviridin, and in that bacteria the reducing power hydrogen do not come from the oxygen, it comes from hydrogen sulfide. Thus oxygen is not produced, sulfur is produced, though the solar energy is utilized for fixing the carbon dioxide, for making the food. Now the next one is the chemosynthetic autotrophs. Chemosynthetic autotrophs, here that is the solar energy is not utilized, that energy comes from certain chemicals. For example, ammonia gets converted into nitrites and then nitrates in certain bacteria, for example nitrosomonas, right? And the energy which is released, that is utilized for the synthesis of the food, so the solar energy is not utilized. That type of autotrophic mode of nutrition is known as chemosynthetic autotrophic nutrition, right? They may be parasitic, as I told you, that is ectoparasite or endoparasite, responsible for developing the disease to the organisms, and in turn they draw the nutrition. Then saprophytic organisms, that is they grow on death decaying organisms. Then for example Clostridium. Then the next one is the symbiotic association. Can you give me one example of symbiotic association? That is a nutrition by symbiotic mode. Symbiotic mode. Any guess? Rhizobium, that is present in the leguminous plant. They form the root nodule and they can provide the nitrogen to the leguminous plant, and in turn they draw the nutrition from the leguminous plant. Any other example? We'll discuss later. You think on it, we will discuss later. Okay, so the mode of nutrition, first is maybe autotrophs, maybe heterotrophs. If it is autotrophs, then photosynthetic autotrophs or chemosynthetic autotrophs. If it is photosynthetic autotrophs, then once again two types, oxygenic and anoxygenic. Then chemosynthetic we have discussed. Then heterotrophic mode of nutrition, that is the parasitic, saprophytic and symbiotic. Right now, the reproduction, they reproduce asexually by binary fission or budding. The structure of the Monera already we have discussed: capsule, pilia, flagellum, cell wall, plasma membrane, and that is the nucleoid single circular DNA. Bacteria can be classified into 4 groups on the basis of the shape. Some bacteria are spherical in structure. They are called cocci. They may be present singly. Then they are considered micrococcus. They may be present in pairs. Then they are termed as diplococcus, or they may be in groups of 4. Then they are termed as tetracoccus, or they may be many in number but remain in chains. Then this association is called streptococcus, or they may be in the form of a cluster and looks like grapes. That type of association is known as staphylococcus. Bacillus, that is the rod-shaped bacteria. They may be with flagella or without flagella. For example, lactobacillus. They may be present singly or in group diplococci, or in chain streptococci, or in a columnar form palisade. 2 other forms, that is the comma or kidney shaped, that is the Vibrio cholerae example, and then spiral shaped. At one end they bear flagella, or may both the ends they bear flagella, example spirillum. So what we have learned? We have learned that is the 4 types of bacteria can be present on the basis of their shape: cocci, bacillus, vibrios, spirillum. Right? This slide represents various types of bacteria like bacillus, spirulina, spiral shaped, staphylococcus. Looks like grape, isn't it? So unique. Now, the kingdom Monera, on the basis of characteristic feature, broadly can be divided into 2 groups. First is Archaebacteria and then the Eubacteria. Eubacteria are considered as true bacteria. In my next video, I will discuss. Example, nostoc. And Archaebacteria, they are the primitive form of organisms. That is, when the earth has originated, they have evolved so they can tolerate high extreme environmental condition, and on the basis of the habitats they are of 3 types: methanogens, halophiles and thermoacidophiles. We will discuss now. I told you just now that the archaebacterians can survive in extreme environmental condition. Can you tell me why? What is the special feature which help them to survive in these adverse environmental condition? That is their cell wall structure, right? Plasma membrane consists of branched lipids along with the hydrocarbon. Outside the hydrocarbon, that is plasma membrane, that is the pseudomuramic acid layer is present, and outside that surface protein is present. These spatial cell wall structure actually help these archaebacterians to survive in extreme environmental condition. First one, halophiles. They are present in the salty region. Then the thermoacidophiles, high temperature, high acidic condition. Methanogens in marshy areas. Let's discuss. First, halophiles. Philes means loving. Halo means in extreme environmental condition. That means they may be present in the high depth of the ocean, and in the deep sea regions where the water is salty. Generally, the organisms cannot survive in hypertonic environment. Why? Because of the exoosmosis, water comes out from the cell and the cells become dead, so they cannot survive. That's why for making the prickle we add salts inside the bottle, isn't it? But these halophiles have special cell wall structure which help them to survive in extreme environmental condition, right? Examples: Halobacterium, Halococcus. Next, thermoacidophiles. Files loving high temperature, high acid loving. That means they can survive in and tolerate high temperature as high as 100 degrees centigrade, and the pH uh, as low as 2, or maybe more than less than 2 also, and thus they can be present in the hot spring region, in the high volcanic region. Example: thermoplasma, thermoproteus, thermus aquaticus. Thermus aquaticus is very important. This Thermus aquaticus is used in biotechnology for PCR, polymerase chain reaction, because in polymerase chain reaction one step is there where the temperature is raised above 90 degrees centigrade, and we know that in high temperature the enzymes disintegrate, isn't it? But as this bacteria Thermus aquaticus present in hot spring region, so that means the enzymes will not disintegrate in high temperature, right? Thus the DNA polymerase, track polymerase, is obtained from Thermus aquaticus, and that is used in PCR. Okay, and here these thermoacidophiles oxidize sulfur to form sulfuric acid, which is the energy which is obtained, that is utilized for the synthesis, chemosynthesis of the food. Now the next one is methanogen. Methanogen present in the marshy region and even in the guts of buffalo, cow, and they can convert carbon dioxide, formic acid, methanol into methane. Okay, that's the reason that from the gobar gas the methane can be produced. Okay, and these methanogens, the examples are Methanococcus, Methanobacterium, etc. Okay, halophiles that I discussed may be purple colored because they contain a special type of pigment like bacteriorhodopsin, but they can solar energy can be converted into ATP, but this ATP is not utilized for the synthesis of the food, so they are heterotrophs. Okay, so now we have come to the end of this video. In this video, we have discussed the basic characteristic features of the monerans, types of bacteria based on their shape, that is the cocci, bacilli, spirillum, vibrio. Then the monerans classification: Archaebacteria, Eubacteria. Archaebacteria, halophiles, thermoacidophiles, and methanogens we have discussed. I hope you have enjoyed learning, isn't it? Enjoy learning and happy learning. Till then, from this side, goodbye.